Hello and welcome to Dr. Varsha Gaitonde's channel. In this video, we shall discuss the current affairs and MCQs continuation in GPB, and this can be named as Part 5. First of all, the evolution of hexaploid wheat, how it evolved, we shall see how many years ago, how the ABD genomes evolved. So, unknown diploid ancestor was there. A and B genome contributors were unknown before few years, but now, as per the genome sequencing and all, everything has been revealed. For the contribution of A genome, Triticum urur 2 is well known. Then for B genome, BB, 2n equal to 14 is the chromosome number. About 6.5 million years ago, unknown wheat-like ancestor gave rise to two diploid lineages, that is genome A and B, which now include Triticum urur 2 and Agilops peltoides. Then these two AABB genomes were hybridized about 8 lakh years ago. So Triticum turgidum was the result, or that is called as wild emmer, having chromosome number 28. AABB is the genome composition. Then the contribution of D genome, that was from Agilops torsi about 5.5 million years ago. Descendants of the two lineages hybridized and produced a new diploid lineage that now includes Agilops torsi or Triticum torsi. From that, about 4 lakh years ago, Triticum turgidum and torsi hybridized and it produced a hexaploid estivum, which is the most widely cultivated species of wheat today. The second topic for the discussion is after GM mustard, GESC, that is Genetic Engineering Approval Committee, set to recommend environmental release of Bayer's herbicide tolerant cotton. As you might be knowing, herbicide tolerant cotton was a big nuisance these days because in states of Telangana and some parts of Andhra Pradesh, without the governmental approval, herbicide cotton, tolerant cotton was in the farmer's field, and that created a big nuisance. Transgenic cotton Bolgard 2 Roundup Ready Flex, that is named as BG2 RRF, contains three alien genes, the first two Cry1AC and Cry2AB, as it was before, being isolated from soil bacterium Bacillus thuringiensis or BT, and coding for protein toxin to the American bollworm, spotted bollworm, and tobacco caterpillar insect pests. This is all about Bolgard cotton, which is already in the market, and that is for pest resistance. The third gene is CP4 EPSPS, is sourced from ancestral soil bacterium Agrobacterium tumefaciens. Its incorporation into cotton makes it tolerant to glyphosate. This herbicide cannot be applied on normal cotton as the chemical does not distinguish between crops and weeds. This particular Bayer's BG2 RRF cotton had undergone biosafety field trials during 2012-13. The dossier containing result of these trials were submitted by its original developer Monsanto to the GEAC in 2013. Amid the regulatory uncertainty and no decision being taken, the American company withdrew its application seeking environmental release of herbicide tolerant GM cotton products. Next is Bayer unveils new hybrid maize for Bihar. So the purpose of discussion of the previous topic was the contributor, the third gene contributor for herbicide resistant. Okay, because you were already knowing the two genes for pest. The third gene was CP4 EPSPS. Okay, and that was initiated by Monsanto and Bayer's. After that, they attempted for herbicide cotton tolerant cotton. Now again Bayer is in news. It unveils new hybrid maize for Bihar. The variety name is DKC, or hybrid name is DKC 9188. Launched a high yielding hybrid maize variety for farmers in Bihar for coming Rabi season. This provides, coming means now already Rabi season is going on. This provides better plant strength and delivers uniform long corn cobs and shiny grains with strong yield stability and higher yield potential. It helps farmers of Bihar to increase the productivity by preventing losses caused by disease and insects. As a part of your assignment, just keep it under the comment section. What are the major pests and diseases of maize? This empowers Bihar progressive farmers to boost their corn yield and improve contribution to India's national crop output. As you know, Rabi maize is very popular and very attractive concept in Bihar because as this was a non-traditional belt of cultivation of maize, now Bihar is widely accepting maize crop in its Rabi season. The third topic is Gujarat to get country's first two organic spices seed parks. Gujarat government released a section letters to two farmer producer organizations or FPOs for the NABARD supported organic spice seed park for SOMP, that is funnel seed, in the Banaskantha district, and Jeera, that is cumin, in Patan for the promotion of organic seed value chain of the spices and to provide capacity building training for the farmers. Total of rupees 23 lakhs in grant assistance given for this and 50 farmers in each FPO. This model will be expanded and replicated in other districts where FPO will oversee the functioning of the parks. Next, we shall just see the current status of area production productivity or the area of particular crops. This is taken from iritoppers.com. Here, for paddy cultivation, West Bengal is the highest producer. This produces about 13.6%, followed by Uttar Pradesh and Punjab. Then for wheat, Uttar Pradesh, Madhya Pradesh and Punjab. Maize, Karnataka, Madhya Pradesh and Maharashtra. Under pulse section, for tur, as you know, tur and gram are the two major pulses of our country. Tur, Maharashtra, Karnataka, Telangana, whereas gram production is highest in Madhya Pradesh, Maharashtra and Rajasthan. Similarly, if we come under commercial crops, sugarcane, Uttar Pradesh, Maharashtra, Karnataka; cotton, Maharashtra, Gujarat, Telangana; jute and mesta, West Bengal, Bihar and Assam. Now we shall see different sectors and its contribution in India's growth. So if we take automobile sector or the particular sector which contributes to India's GDP, 7% share is from automobiles. IT and BPM sector is about 8%. Then construction sector is about 9%. Chemical sector is about 16%. Retail and e-commerce section is about 10%. And the major contributor is food processing sector. Why I discussed about different sectors like automobile, IT, construction, retail, chemical, food processing, because agriculture is the major contributor for our GDP or economy. So 32% share in the Indian market is food processing. That means that comes certainly from the agriculture and its allied sectors. So here 11.6% share is for total employment, 13% share in India's export, 142% cropping intensity is already there. Cropping intensity, you already know how many seasons cultivation is practiced in a year is nothing but cropping intensity. World's largest producer, consumer and exporter of spices. This is all about the data about India. World's largest processor, producer and consumer of cashew nuts. World's second largest producer of food grains, fruits and vegetables. Next concept, this is not related to GPB but crop physiology concept. We cannot ignore our other relevant subjects like the subjects which comes under plant sciences. So this is again an important concept. Do plant need melatonin for a good night's sleep? Certainly. Now the recent research has found this. Melatonin is a natural hormone derived from tryptophan that is, biosynthetic pathways are similar in animals. That means melatonin is also produced in animal biosystem, which causes good sleep and better metabolism. The function of melatonin range from regulation or regulating the circadian rhythms in animals to control the plant growth process from seed germination to leaf senescence. That means from germination to senescence, this particular melatonin is very important. Increase in endogenous melatonin level can be beneficial for human sleep quality and health, as well as for plant field performance, that is better tolerance to environmental stresses. So different functions of melatonin is controlling germination, plant growth and development, regulation of phytohormones and polyamines biosynthesis, then antioxidant activity and reactive oxygen species scavenging, then tolerance against abiotic and biotic stresses. One more concept is one nation one fertilizer. As you are well known that many of our private companies or government undertaken mini ratna maharatna projects were taking or importing the fertilizers from different countries and were selling in different brand names. So farmers were in confusion which one to take, and there was a major havoc in the market because of the fake fertilizers produced by the fake people. So to avoid all those things, government has brought a big initiative with the brand name of Bharat. Whichever company it may be, fertilizers are sold under the title of Bharat on subsidized rate. According to One Nation One Fertilizer order, irrespective of the company that manufactures it, whether in the public or private sector, or fertilizer bags, all fertilizer bags, whether containing urea, DAP, MOP or NPK, will sport the brand name Bharat Urea, Bharat DAP, Bharat MOP, Bharat NPK, like that, so on and so forth. Our last topic of the discussion is Punjab to supply paddy straw to fodder shortage hit in Kerala. Okay, that means, as you know, every beginning of every winter season there will be havoc or a big problem in Delhi and NCR region about the environmental pollution. So this is mainly due to the burning of paddy straws and other raw materials in the field. So, as you know, southern states are mainly concentrating on milk production, but due to certain things, the hay or the straw supply and availability is less for the cattle, whereas Punjab can supply more amount of these things. So, Punjab has agreed to supply paddy straw to Kerala for use as fodder for livestock, as the dairy sector in the southern state faces an acute shortage of green fodder and hay. The two states have agreed to transport paddy straw to Kerala using Centre's Kisan Rail scheme. As you know, for better transport of the agriculture, perishable goods, and everything, Kisan Rail project has been introduced by the central government 2 or 3 years back. The initiative is seen as a win win situation to both states. While Kerala gets much needed fodder, it will help Punjab deal with excess paddy straw, which contributes to stubble burning and air pollution. The dairy sector in Kerala, second only to Punjab in milk production, is a means of livelihood to lakhs of farmers. However, acute shortage of quality cattle feed and relatively high prices are the major hurdle. So, cooperative federalism works here, and because of that, the two states are happy, and even the milk production in Kerala can also be increased. So, come to the MCQ section. The first question today, we shall discuss some of the biotechnology related questions. The first question is, in which year Maxim Gilbert gave technology of DNA sequencing? Maxim and Gilbert, as you know, they are the initiators of DNA sequencing technology. It's popularly known as Maxim Gilbert technology. So they proposed during 1977. The options are 1977, 1987, 67, and 57. Answer here is 1977. Okay. The second question is, bialistic gene transformation method for plant transformation was developed by. The four options are Burton, Klein, Williams, and Smith. Here, bialistic gene transformation, there are different types of gene transformation, and under that, bialistic with the gun, this technology is used. The process was given by Klein. The options here are Borton, Klein, Williams, and Smith. Answer is B. Bialistic gene transformation method was developed in the year options 1987, 1990, 1995, and 2000. Answer is 1987, that is A. In which year International Rice Genome Sequencing Project was completed? Options 2000, 2005, 2010, 2015. Answer is 2005. Bt gene is isolated by, the four scientists' name are given here: Borton, Fodder, Williams, and Powell. The answer is Borton and co-workers. Option A is the correct answer. Next question is, biotechnology is the dash use of biological agents. Options are open, controlled, accessible, and limited. It is controlled use because small mistake may also cause severe environmental havocs. So controlled use is the correct option. The chemical used to protect cell or tissue against freezing injury. As you know, freezing injury is the major problem under tissue culture. So there are four options given: NH4, methyl sulfonate, dimethyl sulfonate, and DMSO. The chemicals used in tissue culture and its purposes are discussed in detail in a separate video, and already available in this channel. So the answer here is DMSO. Next question, progeny generated or regenerated from tissue cultured plantlet is denoted as. Answer is R1. Options are R0, R1, R3, R2. R stands for regeneration. Okay, so progeny regenerated from tissue cultured plantlets. The tissue cultured first generation plantlets are R0, means from where the regeneration is taken is certainly R1, and subsequent generations will become R2, R3, and so on. Next question, agar is isolated from options bacteria, yeast, weed, and algae. The answer here is algae. As you know, it is a seaweed. Next question, the most common carbon source used in plant tissue culture media is options fructose, sucrose, glucose, and galactose. Generally, sucrose is used because it's a reducing sugar. Again, after breaking, it will become glucose. So whenever ready-made source is required, glucose is used, because but in most common purposes, sucrose is the major source used in media. Now here I will give you an assignment. You should tell me why sucrose, why not the other sugars used in tissue culture media. Okay, keep it under comment section. Next question, shoot proliferation from callus is promoted by. Options IAA bar NAA, BAP bar BA, then GA3 and ABA. As you know, the first is auxin, second is cytokinin, GA3 is gibberellin, ABA is abscisic acid. Okay, so for root proliferation, auxin is used. Shoot proliferation, cytokinin is used. So here for promotion of callus, what shoot proliferation, what is used is the question here. As you know, certainly BAP or BA is a cytokinin source. That is the answer. Next question, which of the following is the thermolabile compound? Thermolabile means the compound which is labile, or it may become melt, it may melt or volatilize due to thermal treatment or by high temperature. Options given here are plant growth regulators, agar, charcoal, and micronutrients. Option here is A, that is plant growth regulators. This is the region where the plant growth regulators are not kept in autoclave for autoclaving. Okay. Next question, which of the following is not a plant tissue culture nutrient media? Options are MS, Gamberg, B4 and B5 and S9. We have already heard MS, Gamberg, B5 are already different types of medias and used for different purposes, whereas S9 is not any media or plant tissue culture media. Okay, answer is S9. The formation of embryo from callus is known as options parthenogenetic, indirect embryogenesis, direct embryogenesis, none of these. Here you should know what is direct embryogenesis and indirect embryogenesis. In case of direct embryogenesis, there won't be formation of callus. Okay, before embryo formation, if callus is formed means that is indirect embryogenesis. Okay, so here what they have asked, formation of embryo from callus means callus is here. Now one tissue is taken, then callus formation will happen, then somatic embryo is formed. Okay, if it is present means that is indirect embryogenesis. Option B is the correct answer. Next question, the development of a structure from an unrecognized state to differentiate and organized state is. The options given here are redifferentiation, totipotency, morphogenesis, all of these. I'll read out the statement once again. Development of a structure from unorganized state to differentiate and organized state. The answer here is redifferentiation. But before knowing that, you should know what is the morphogenesis, differentiation, de-differentiation, redifferentiation, all these definitions. Okay, totipotency is ability of any tissue to become a complete plant. Okay, morphogenesis means ability to get into different morphology. Okay, now we shall see the definition of differentiation, what is de-differentiation and redifferentiation. Okay, differentiation is where the plant cells will form into shoot apex, root apex, cambium, meristem, means different meristematic tissues are produced from cells is the process, nothing but differentiation. Now what is de-differentiation? It is the process by which structures or behaviors specialized for a specific function lose their specialization and become simplified or generalized. That means it will again become a cell. Okay, whereas redifferentiation is process by which group of once differentiated cells return to their original specialized form. Okay, other differences are also given here, you just refer it. Next question, for filter sterilization, pore size should be. Options are less than 0.45 micron, more than 0.45 micron, more than 0.45 mm, or more than 25 mm. Here the answer is less than 0.45 micron. Okay, why? Because if more pore size is there, tissues or cells will drain away. Next question is, for explant sterilization, dash percent mercury chloride concentration generally used. Options here are 0.1 to 1%, 5 to 10%, 0.0001 to 0.01%, 20 to 25%. The answer here is 0.1 to 1%. Okay, that much mercury chloride, because higher concentration of mercury chloride will become toxic to the cells. Next question, which of the following chemical not used for sterilization of explant? Here not used. Okay, options given are bromine water, H2O2 or hydrogen peroxide, HgCl2 or mercury chloride, then Si. Bromide water, H2O2 and HgCl2 all are used for sterilization of explant, whereas silicon is not used. As you already know, silicon is used in our computers, mobile phones and all for production of chips and all. That's a different story. Next question is, dash percent of ethanol is used for sterilization of explants. This is universal, that 70% is the percentage used in sterilization. Next question, nutrients required dash concentration is also known as macronutrient. Options are more than zero point 0.5 micromole per liter, more than 0.005 micromoles per liter, less than zero point 0.5 millimoles per liter, or less than 0.005 m moles per liter. The option here is, the answer here is more than zero point 0.5 m mole per liter, that is macronutrient. Polarity of somatic embryo is, somatic embryo, as you know, after getting tissue will treat with certain media and will get the somatic embryo. Here the options are multipolar, monopolar, bipolar, all of this. Somatic embryo, after the generation of embryo, it will produce root and shoot and root. Okay, so it is bipolar because shoots are also coming, roots are also coming, so two polarities there, bipolar. Next question, embryo rescue technique helps to overcome. The answers are post fertilization barrier, vitrification, browning problem, and pre fertilization barrier. Embryo rescue technique is used whenever there is problem with post fertilization barriers, because here we are using the rescue technique of embryo. Okay, embryo is formed after the fertilization only, so it is post fertilization. Before that you should get to know what is pre fertilization barrier and what is post fertilization barrier. In pre-fertilization barrier, when these barriers will come, whenever you are going for interspecific or intergeneric hybridization. Okay, so failure of zygote formation, failure of pollen germination on stigma, insufficient growth of pollen tubes on style, or cross incompatibility are some of the pre-fertilization barriers. Whereas for post-fertilization, failure of zygote development, hybrid sterility, hybrid breakdown in F2 or later stages are the main reasons. So for that, embryo rescue technique is used as a post-fertilization barrier. Okay. Now, I have left one concept, that is similarity between dedifferentiation and redifferentiation. As I have already discussed about the differences, but here similarities are also very important because this is asked repeatedly in many of the competitive exams. So you should definitely know the three things: differentiation, dedifferentiation, and redifferentiation. Okay. So, the similarity is cells change their ability to divide through two mechanisms, that is dedifferentiation and redifferentiation. Both process occur in differentiated cells. Both systems are also important in the genesis and healing of damage. Next question, seed propagation of sterile plant is possible through. Options are pollen culture, ovary culture, in vitro fertilization, and synthetic seed. What is the question? Seed propagation of sterile plant. Okay, sterile plant means there is neither production of male part or female part. So pollen culture means male culture, that is already absent, ovary is already absent. So when those two are not present, in vitro fertilization is waste process. So synthetic seed, where the somatic embryo is encapsulated and used, is the option. In somatic embryo production, what they'll take any of the asexual part or asexual cell or tissue. Okay. Next question is, high sucrose level acts as dash in pollen culture medium. Options are diffusion, plasmolytic, osmolytic, osmoregulation. The answer here is osmoregulator. Okay. Sucrose acts as a osmoregulator. Next question, hyperhydration or vitrification problem is associated with. Options are micropropagation, pollen culture, embryo culture, or ovule culture. Answer here is micropropagation. Okay, in micropropagation, hyperhydration and or vitrification is the major problem. Next question, induction phase of embryogenesis is promoted by. There will be different phases in plant tissue culture. You should know it. First one is lag phase, then log phase, then this induction phase, and finally the death phase. Four phases will be there in a tissue culture development curve, growth curve. So here four options are given: auxin, ABA, cytokinin, and GA3. For induction phase, auxins are important, or this promotes the induction. Next question, for embryogenesis, which of the following developmental events are in correct sequence? So the correct sequence is asked. I'll just give you the answer here, because if I'll read out all the things, you will get confused. The correct option is B, that is first globular form will come out, then it will become heart shaped, then torpedo shaped, then cotyledons will come. Okay, so globular, heart shape, torpedo, cotyledonary is the correct answer for embryogenesis. Next question, gynogenesis term related to. Options: fertilized ovule, unfertilized ovule, mature ovule, both A and B. Gynogenesis is what? It is about female reproductive part, genesis of the female part, that is related to unfertilized ovule. Okay. Next, in which phase the rate of cell division is highest? As I have already given you the growth curve in tissue culture, first lag phase, then log phase, then exponential phase, and decline or death phase. Okay, so during exponential phase, here the cell expansion takes place. Next question, in which phase both the rate of cell division and expansion are decline? Decline means it is linear phase or death phase. This is the linear phase. Okay, in pollen culture medium, high sucrose level may act as. Options are osmoregulatory, plasmolytic, osmolytic, and diffusion. Here, as we discussed earlier also, it acts as a osmoregulator. Okay, sucrose acts as osmoregulator, means keeping the osmotic potential balance. Next question, the technique which used to recover hybrid from sexually incompatible species. Options are in vitro fertilization, in vitro pollination, embryo culture, unfertilized ovule. The answer here is embryo culture. In vitro means inside the test tube the fertilization is done. Embryo culture, as you know, embryo are taken and rescued. Unfertilized ovule, here it is not used. Next question, pre-fertilization barriers in wide hybridization can be overcome by. As I have already explained pre-fertilization and post-fertilization barriers in previous slides. The options given here are embryo culture, embryo rescue, in vitro pollination, and anther culture. The answer here is C, in vitro pollination. Now, the tendency of callus cells to separate easily is known as. Callus is undifferentiated or unorganized mass of cells. Here, options are friability, hybridity, callusing, or granulation. Callusing is the formation or becoming solid. Okay, hybridity, as you know, hybridization or crossing between two or three. Granulation is granule formation. Callus cells to separate easily is nothing but friability. Okay, the answer here is A. Next question, regeneration of plants from tissue to of actively dividing organs like stem tip. Options: shoot culture, meristem culture, organ culture, cell culture. Okay, the answer here is meristem culture. Meristem is nothing but the dividing cells. Next question, production of haploids can be done by. Options: chromosome elimination, chromosome doubling, double fertilization, all the above. Answer here is chromosomal elimination, because by chromosomal doubling we cannot go for hybrid haploid production. Double fertilization also won't yield haploids. Only chromosomal elimination technique will help in haploid production. Next question, in which phase cell attain capacity for embryogenesis and develop up to globular stage? I have already told you there will be globular, heart, then torpedo, and cotyledonary stages. Okay, so for globular stage, Induction phase, that is option B, will give the capacity for embryogenesis. Okay, this induction phase is the answer, that is the third phase. Next question, production of haploid embryo of plantlets from pollen grains is known as you know, pollen grain is a male part. Okay, androgenesis, androgenetic, gynogenesis, both A and B. Androgenesis is the answer here. Which of the following hormone used for maturation of somatic embryo? The straight answer is ABA. Maturation and senescence means this ABA and ethylene will work. So ABA is the answer here, option A. Next question, generally pH of media is adjusted to 5.8 always. Okay. When somaclonal variation is not heritable, it is called as, answer is epigenetic. This is a straight answer. Epigenetic is the answer here. Okay. The basis of somaclonal variation is karyotype change, transposable element, gene mutation, all of this. Somaclonal variation happen by all these three means, that is karyotype change, transposable element and gene mutation. Next question, fertilized ovule culture will produce, options haploid, diploid, triploid and pentaploid. Ovule culture, fertilized ovule means it is already in diploid condition, so diploid is the answer here. Encapsulation of somatic embryo is required in case of, encapsulation, as I have already told you, this process is done with sodium alginate or calcium alginate, generally it is preferred. With gelling agent, encapsulation is done for somatic embryo, that is done under synthetic seed. The encapsulated somatic embryo is, options are capsulation, synthetic seed, somatic seed, all the above. Synthetic seed is the answer here. The concept of synthetic seed was developed and term used by, options are Toshiba, Morishige, Smith, Nathans, Maheshwari et al. And co-workers. The option is Toshiba, Morishige. Okay, Morishige is the answer. Artificial seeds may be produced by, the different ways are desiccated, hydrated system, none of this, both A and B. Artificial seeds are both desiccated and hydrated, means dry seeds as well as wet seeds will be there. Dry will be more preferred. Next question, who used enzyme for protoplast isolation for the first time? The options are Takebe, Cocking, White or Kranz. As you know, White's media used to be there, Kranz anatomy was given by Kranz. Then Cocking is the answer here, where the protoplast isolation first time who used the enzyme in protoplast isolation. Next question, the cell divide of cell wall and nucleus is known as, option cytoplast, protoplast, cytome and chloroplast. Okay, so here you should know these individual terms. Protoplast, as you know, the outer covering of any plasma. Protoplast is the, without cell wall, it is known as protoplast. Then cytoplast is the answer here, where the cell divide of cell wall and nucleus. Chloroplast, as you know, it's organelle present in our cell system. Cytome is the cellular system where structural and functional heritability of an organism is defined, that is cytome. Next question, in case of protoplast fusion, which of the following stages are in correct sequence? Options 4 are given: agglutination, membrane fusion, rounding off of protoplast. Option A is the correct answer. Here other things I am not going to read because they may confuse you. Okay. Next question, application of electric current for protoplast fusion is known as, options microinjection, electrophoresis, electroporation, none of this. The answer here is electroporation, C. Spontaneous fusion of protoplasts often produce, options homokaryon, heterokaryon, hybrid, both A and B. Answer is A and B, because by spontaneous fusion of protoplasts, either it may give homokaryon or heterokaryon, that means similar fusion or differential kind of fusion. Next question, pectinase enzyme is isolated from Aspergillus niger. This is a straight question. Meserozyme is isolated from Rhizopus species. Somatic hybrid with normal protoplast of one species and nucleus less protoplast of other species. This is a definition of hybrid. Okay, option B is the answer here. Next is the cellulose enzyme derived from. Cellulose is always derived from fungus. Option A is the answer there. Which of the following are used as osmoticum? Options are sorbitol, mannitol, glucose, all of this. Answer here is all of these. Osmoticum added in protoplast isolation for. Options are to prevent bursting of protoplasts, maintain osmotic pressure, then increasing osmosis, reducing osmotic potential. Here, sorry for the inconvenience, already answers are ticked here. The answer is to prevent bursting of protoplasts. Next question is hybrid is produced due to partial hybridization is asymmetric hybrid. Here the question asked is partial hybridization means asymmetry is found here. Next question, hybrid which developed through fusion of two normal protoplast of different source. In the previous this one also we discussed the same thing. Here definition is somewhat different. See, somatic hybrid with normal protoplast of one species and nucleus less protoplast of other. Okay, the same is here. Fusion of two normal protoplasts of different sources is asked. That is nothing but hybrid. Next question, restriction endonuclease having identical recognition sequence but different cleavage sites are called. Answer is straightforward, neosesomers. Okay. Next question, which of the following is DNA modifying enzyme? S1 nuclease, cellulase, protease and RNase are the options. Here the function of cellulase is to cleave the cellulose or to degrade the cellulose. Protease is to dissolve the proteins. RNase for against RNA. S1 nuclease is for DNA modification. Last part, widely used restriction endonuclease for gene cloning is. Here, before getting into these questions, we should know what are restriction enzymes and what are the different types. Restriction enzymes are the enzymes which cut the DNA at specific sites. So, what is the purpose? Restriction enzymes are basically used under gene cleaving, cloning, marker assisted selection, then association mapping and all those studies of markers as well as in cloning technology. So there will be three types of restriction enzyme: type 1, type 2, type 3. Type 1 enzymes, this cuts the DNA far from recognition sequence. No much practical value is found for type 1 restriction enzymes, whereas type 2 cut at specific position closer to the restriction site, which produce blunt end and sticky ends. These are generally used, means initially type 2 enzymes were used. Then type 3 are multifunctional proteins with two subunits, that is for restriction and modification. So widely used restriction endonuclease for gene cloning is type 2. Okay. Next is HIND3 is belonging to which category of restriction enzyme? Again, HIND3 is type 2 restriction enzyme. E. coli is a dash type of restriction type 2 restriction enzyme. Last two questions are from plant breeding. Baby corn is harvested at the stage of, answer is 2 to 3 days after tasseling. Last question, which among the following is a bunch type of variety of groundnut? As you know, there will be spreading type and bunch type of varieties. AH334, RG382, RG425 and Girnar 2. Here the answer is Girnar 2 is the bunch type of this one groundnut variety. Okay, so friends, if you have any doubt or any specific topic to discuss, just keep it under the comment section. Thanks for your patience watching. Keep on sharing, subscribing and commenting. Thank you once again.